Assalamualaikum and hi everyone. I'm Nur Hazlini Matzin, postgraduate student from University Malaysia Kelantan. My slide presentation entitled Assessing the Floral Volatile Constituents of Male and Female Rafflesia kerri Meijer from Lodging Highland Peninsula, Malaysia. Without further ado, let's take a look at my slide. Content consists of introduction, general background of Rafflesia, problem statement, significance of study, objective, methodology, result and discussion, conclusion and recommendation, references and the acknowledgement. Rafflesia around the world. Rafflesia is a genus of parasitic flowering plant from the family of the Rafflesiae, distributed only in tropical rainforests of Southeast Asia country, which includes Thailand, Indonesia, the Philippines, Malaysia, and Brunei. Here is the map of distribution of Rafflesia in Malaysia, where 5 species reported from Sabah and Sarawak, while 7 species reported from Peninsular Malaysia. Here is my subject study Rafflesia kerri Meijer. In Peninsular Malaysia, Rafflesia kerri was nominated as the largest species. The species was named after E.F.G. Kerr, who discovered the flower in Thailand 1929. In 2010, a scientific expedition organized by UMK had identified 26 hotspots of the flower in Lodging Highland. Thus, this flower was used as the icon for ecotourism in Lodging Highlands. The place mostly was visited by the international tourists. However, due to global pandemic COVID-19, the activities for foreign visit was stopped. As an odoriferous flower, Rafflesia was pollinated by calipharid flies, and Rafflesia kerri was found to parasitize on Tetrastigma rafflesiae and Tetrastigma hookerii. What issue arise regarding Rafflesia? Rafflesia is a rare flower. Several natural factors contributed to the rarity of the Rafflesia flower. Not only that, the human activities also contribute to decreasing number of Rafflesia population and thus lead the species to the brink of extinction. This is a very serious issue that every people need to take part, including government, NGO, and us, the community. So how to overcome the problem? A successful pollination process resulting to fruit formation contains thousands minuscule of seeds. So for the pollination process to occur, we need to understand what is the key that attract pollinator to allocate and visit the flowers. Exactly, the fluorescence. The scent emitted during the anthesis stage acts as chemical cue, luring the pollinators to visit the flowers. However, the knowledge regarding the fluorescence is still limited and scanty. So many questions were left without an ample answer. So the problem statement, we want to know where is the fragrance come from? What is the scent emitted during the blooming stage? Which chemical constituent responsible in attracting the pollinator to visit the flowers? Therefore, the objective of this study to evaluate the presence of chemical compound through the qualitative phytochemical analysis and screening the floral volatile constituents. The impacts of this study fill in the gap to our knowledge and also give the impact to the society and country. Rafflesia is rapidly becoming a prominent symbol for conservation efforts. All the conservation works to conserve the flower and directly conserve our tropical rainforests. The knowledge transferred through direct field work help the orang asli to work as tourist guide, creating job opportunity for them, and thus they have an alternative income to support their living. This ecotourism sector are able to boost our economic growth. Local tourists should support our tourism sector. But I need to stress out here, a proper plan such as sustainable ecotourism should be adopted so the place will protected from human intrusion. Methodology. The field sampling was conducted in Rafflesia conservation area. Male and female flower were chosen due to different in structure as shown in plate B and plate C. Eternally extract preparation. The collected sample were weighted and cut into small pieces to allow more chemical release to the solution. The samples were macerated at room temperature for 3 days. Figure G shows the extract obtained. The eternally extract were labeled with MCD for male flower and FCD for female flower. For qualitative phytochemical screening of MCD and FCD, methods is based on Harbin 1984, whereas for floral volatile constituent screening, 2 fibers were selected, known as PDMS and DVB carboxylane PDMS fibers. Prior to operate, all fibers conditioned at 250 degrees Celsius for 2 minutes. About 0.5 grams of each eternally extract was placed into 20 ml vial and tightly capped. The vials were introduced in a thermostatic bath 60 seconds at 50 degrees Celsius and vibrated for 5 seconds every 2 seconds. After sampling, the fibers was immediately inserted into the GC injector for 1 minute. The volatile constituents were qualitatively identified by matching the mass spectrum with database Wiley and HPCH library. So, here is the result and discussion for the test conducted. The result for the qualitative phytochemical screening. From table 2, we can see most of the tests give the positive result except for saponin test. For alkaloid test, the white precipitated was formed at the bottom of tube. Greenish blue color was observed, indicates the presence of steroid, whereas for flavonoid test, the test extract solution changed the yellow color, and for tannin test, the extract solution changed to blue-black color precipitate. Roles of secondary metabolites in plants are for alkaloids, end product of metabolism, source of nitrogen, plant defense, growth regulator, and alternative source of minerals in plant. Sofian T. 2008 reported on Rafflesia hassel tea where 2 alkaloids compounds, nicotine and caffeine detected. The presence of this compound possibly influenced the behavior of insect towards the flowers. 3 terpenoid steroids was recognized to have potential as sex hormone, anti-tumor, plant growth hormone regulator, and antibacterial properties. Whereas flavonoids are believed to give color to Rafflesia visual displays, whereas terpenoids, amines, and phenylpropanoids contribute to scent mixture, luring pollinators to visit the flower and thus assist in pollination process. Tannins play role in plant defense activities by act as antimicrobial against plant pathogens, and as the astringent taste of tannins help in feeding repellents of herbivores. For floral volatile constituent screening, the data obtained showed more peaks detected from PDMS fibers compared to gray fibers. This indicates that the majority of the VCs are from the non-polar compound. In MCD eternally abstracts, about 45 of VCs were detected by using PDMS fibers, whereas 16 VCs detected using gray fibers. In FCD eternally abstract, about 68 peaks were detected by using PDMS fibers, whereas 5 VCs were detected using gray fibers. Plant emit variety of volatile organic compound as response to the biotic and abiotic stresses. The VOC have chemical properties such as low molecular weight, high vapor pressure, and low boiling point. Floral volatile constituents play a crucial role in pollination biology by emitting a strong scent, luring pollinators to visit the flowers. Other than serve as attractant, the FVCS serve in defense system against the herbivores and other plant pathogens includes bacteria, virus, and fungi. Long-chain hydrocarbons, which include alkanes and alkenes, are the common compound found in plants and insects and play a role as male sex pheromone in many species. Seashell 2008 reported the flower that produce alkenes responsible in the attraction of the pollinator behavior. Thus, the composition of fluorescence with the presence of long-chain hydrocarbon group believed to attract pollinators and other visitors to locate the Rafflesia and increase the pollination success rate. Organosilicon group is colorless, odorless, lightly volatile, and widely used in cosmetics, plastic, and hygiene product. The presence of this compound in the extract believed to give the viscous resin-like characteristic to the extract. The VCs from this group believed to be valuable than the monoterpene hydrocarbon due to contribution in the fragrance of the essential oil. 2-ethyl-1-hexanol is mild, oily, sweet, and has slightly rose floral odor. This VC also was identified in Rhizanthus lowi, where Rhizanthus lowi attracting the same calyphorid flies as Rafflesia. Besides, 1-octadecanol not only identified in plant extract but also the hair pansa glands of males of Heliotis virescens from Lepidoptera, which can explain to us of how plant insects communicate and able to locate the host plant. Aromatic acids. 1,2-benzene decarboxylic acid dialkylate ester has a very slight and aromatic odor. This constituent was identified in apricots and plums. This finding support Bazinga's statement when he explained the weaker fruity fragrance similar to dry apricot and peaches comes from the tube cavity. Besides, 2,4-D-isocyanato-1-methylbenzene is a chemical with sweet, fruity, and pungent odor and was found in the feces, urine, and tissue of carcass, which possibly contribute in attracting the carrion flies to visit Rafflesia. William 2090 discovered 13 tetradicenol acetate constituent as sex pheromone to attract the male click beta melanotus communities. The presence of these constituents blend in with other compounds was likely being the reason for other visitors to visit Rafflesia. 3 VCs were grouped into miscellaneous compounds. The information regarding all of these VCs were limited and thus need a further study. To sum up this presentation, Rafflesia contain alkaloids, styroflavonoids, and tannin as plant secondary metabolites, which play a role in defense system, antimicrobial, antioxidant, nitrogenous storage, attractant to pollinators, and repellent against plant herbivores. During blooming stage, Rafflesia emit the combination of chemical compound, for example, primary alcohol, aromatic acid, long-chain hydrocarbon, organosilicon compound, and also other miscellaneous compound. This combination of chemical was believed during the insight to locate and visit the flowers. More research activities recommended to be conducted in competitive studies such as identification of collected insect specimen into species level, identification of the unknown volatile constituent, and field tests to determine insect activities and response toward the identified FVCS. This is the list references for the study. Last but not least, this research was fully funded by Research Grant Ministry of Education. Also, special thanks to UMK and my supervisor Zulhazman Hamzah. Thanks to Puan Mailina and other research team from Forest Research Institute, Majlis Daerah Gua Musang for allowing me to conduct the research in Rafflesia Conservation Area, and I'm also grateful to Pejabat Tanah Lojeng Highland for providing me a place to stay. Thank you to Citra Secretariat for making this conference possible. Also through virtual presentation, thank you so much for your kind attention till end of my slides. Hopefully everyone doing good and stay safe. Don't forget to follow my IG Norhazline MZ. Hashtag kita jaga kita, stay home, stay safe, COVID-19, and surely hashtag Citrus 2.0. Thank you.